Imagine a bustling city. Buildings tower overhead, streets teem with activity. This is a city that never sleeps. Now imagine something much smaller, yet just as complex. This is the world of the cell, the fundamental unit of life. Just like a city comprises countless buildings, our bodies are made up of trillions of cells. Each cell is a tiny, self-contained unit, working tirelessly to keep us alive. Cells are responsible for everything we do. They provide structure and support. They convert food into energy. They transport nutrients and remove waste. They even defend us from harmful invaders. Without cells, life as we know it would not exist. From the simplest bacteria to the largest animals, all living organisms are made up of cells. Understanding how cells work is fundamental to understanding life itself. It allows us to diagnose diseases, develop new treatments, and unravel the mysteries of the natural world. This journey through the cell will reveal the intricate workings of these microscopic marvels. We will explore their structure, their function, and the remarkable processes that keep them going. The world of cells is surprisingly diverse. Cells come in all shapes and sizes, from the long, slender neurons that transmit nerve impulses to the disc-shaped red blood cells that carry oxygen throughout our bodies. However, despite their differences, all cells can be broadly classified into two main categories: prokaryotic cells and eukaryotic cells. Prokaryotic cells are the simplest and most ancient form of life. They are smaller and less complex than their eukaryotic counterparts. Bacteria and archaea are examples of prokaryotic organisms. These single-celled organisms thrive in a wide range of environments, from the depths of the ocean to the human gut. Eukaryotic cells, on the other hand, are larger and more complex. They are characterized by the presence of a true nucleus, a membrane-bound compartment that houses their genetic material, DNA. Animals, plants, fungi, and protists are all examples of eukaryotic organisms. One way to think about the difference between prokaryotic and eukaryotic cells is to imagine a one-room apartment versus a multi-room house. The prokaryotic cell, like a studio apartment, has a simpler structure with fewer internal compartments. In contrast, the eukaryotic cell, like a house with multiple rooms, is more complex and organized, with specialized compartments for different functions. Every cell, whether prokaryotic or eukaryotic, is enclosed by a thin, flexible barrier called the plasma membrane. This membrane acts as a gatekeeper, controlling the movement of molecules into and out of the cell. It is selectively permeable, meaning that it allows some substances to pass through while blocking others. The plasma membrane is made up of a double layer of phospholipids, a type of fat molecule. These phospholipids are arranged in a specific way, with their hydrophilic, water-loving heads facing outward and their hydrophobic, water-fearing tails facing inward. This arrangement creates a barrier that is impermeable to most water-soluble molecules. In addition to the plasma membrane, some cells, like plant cells and many bacterial cells, also have a rigid cell wall surrounding their plasma membrane. This wall provides structural support and protection to the cell. It helps maintain the cell's shape and prevents it from bursting under pressure. The composition of the cell wall varies depending on the organism. For example, plant cell walls are made primarily of cellulose, a complex carbohydrate, while bacterial cell walls are made of peptidoglycan, a polymer of sugars and amino acids. The inside of a cell is a bustling hub of activity. It is filled with a watery, gel-like substance called cytoplasm. This cytoplasm is not just a passive filler, it is a dynamic environment that plays a vital role in many cellular processes. It provides a medium for chemical reactions to occur and acts as a solvent for various molecules. Embedded within the cytoplasm is a network of protein fibers called the cytoskeleton. The cytoskeleton provides structural support to the cell, much like the frame of a building. It helps maintain the cell's shape, allows it to move and change shape, and plays a crucial role in cell division. The plasma membrane, with its embedded proteins and other molecules, is often described as a fluid mosaic. This term reflects the dynamic nature of the membrane. The phospholipids and proteins are not static. They are constantly moving and shifting within the membrane, like a mosaic of tiles that can rearrange themselves. The plasma membrane's selective permeability means that cells have to be picky about what they let in and out. Essential nutrients need to enter the cell, while waste products need to be removed. To achieve this, cells have developed various mechanisms of membrane transport. These mechanisms can be broadly categorized into two main types: passive transport and active transport. Passive transport does not require energy. It relies on the natural movement of molecules from an area of high concentration to an area of low concentration, a process known as diffusion. One common example of passive transport is osmosis, the movement of water across a semi-permeable membrane. Active transport, on the other hand, requires energy to move molecules against their concentration gradient, from an area of low concentration to an area of high concentration. This energy is usually supplied by ATP, the cell's primary energy currency. Active transport is essential for cells to maintain the right balance of ions and molecules inside and outside the cell. One example of active transport is the sodium-potassium pump, which helps maintain the electrochemical gradient across nerve cell membranes, crucial for transmitting nerve impulses. Some cells are not content to simply sit still. They are capable of movement. This movement is often facilitated by specialized surface appendages, such as cilia, flagella, and pili. These structures extend from the cell surface and help cells move through their environment or move fluids and particles past the cell. Cilia are short hair-like structures that move in a coordinated wave-like manner. They are found on the surface of many cells, such as those lining the respiratory tract, where they help sweep mucus and trapped particles out of the lungs. Flagella are longer, whip-like structures that propel cells through their environment. Sperm cells, for example, use their flagella to swim towards the egg. Pili are shorter and thinner than flagella, and they are often involved in attachment to surfaces or other cells. For example, bacteria use pili to adhere to host cells, a crucial step in the process of infection. In some cases, pili can also be involved in a type of bacterial conjugation, where genetic material is transferred from one bacterium to another.